Thanks a lot, Gina, for the invitation. And it's been, I guess, maybe all your speakers tell you this, but it's been super fun. I've really enjoyed chatting to everybody here. It's been a really, really fun department, and I've really, I feel like the research here is so exciting. Personally, it's exciting for me. I grew up not far from here, and for some reason, as an undergrad, I chose not to go here, but my next two brothers did, and they both, I think, worked with John. Or did Oren work with you, Tim? No. I think they both worked with John Vandermeer here as undergrads. So it's fun to come back come here and talk to you all. And so, as Gina said, I'm really interested in how plants mate, how they make these mating decisions. And I was meeting with Nia today, one of Gina's students, and she said, "Have you always cared about this?" And I said, "I can't tell if I even care about this today." Okay? And so the reason why is I don't think that you have to come here saying, my motivating question is to learn about how plants mate to get something out of this talk. The reason I care about this question about how and why plants make their mating decisions is because I think it has a lot of implications for how we think about biology more broadly. Okay, so I guess you're here, so I don't have to sell you. You're stuck here for the next, I don't know how long, but the point is that you don't, if you're interested in animals, if you're interested in phylogeography, if you're interested in these bigger questions, I think this is a great way to try to address these questions. So just by way of introduction, I am interested in what I think are some of the bigger problems. I guess we all think we're interested in big problems, but the big problems in population genetics and where micro and macro evolution meet. So how is genetic variation distributed? What's the nature of that variation? How does that what are the processes that generate and structure it? How do we go from small scale evolutionary processes to larger scale patterns that we see in the world? So these are some of the motivating questions, and this question about who mates with whom is how I get at this. And I think it's because it leverages lots of these variables that you might want to look at. So for example, if you're interested in the response to selection or the importance of epistasis, when you co-inherit a lot of your genome because you're selfing, all of a sudden different components of genetic variation are available for selection that usually aren't seen when you're selecting in a large panmictic population. So you can compare the response to selection in a selfing species versus an outcrossing species, and you can learn about the nature of variation. I'll tell you this as we go. It's okay. And so I try to bring in diverse data sets to answer these questions. And I try to do my work, my empirical work, strongly related to theory, and theory strongly related to empirical work. So I think that's the real way that I find to make progress in that. And that also means that I have to collaborate often. So I think it's hard for theorists to do a good job on their own, or empiricists to do a good job without the hand of theory. So I often find if I'm working on a theory problem where I'm not as good as a theorist, I'll collaborate with a theorist. If I find an empirical problem where I need good field data, I can't do that, and so I need help. Okay, so I'm going to talk a little bit about variation in plant mating and some of the implications. I'm going to talk about some of my older work and work by others and some of these ideas. I'm going to then talk about some consequences of how plants mate, and specifically thinking about where we find them, where we find different plant species, and then a role on hybridization and introgression, largely in plants, and the role that plant mating system might have in this. And then some theoretical work about tensions over mating decisions. Okay. So the first thing I want to say is, as I said in the introduction, there's a tremendous amount of variation in how plants mate, and this allows us to test some of the basic theoretical questions that we're interested in. And when I think about how plants mate, I'm thinking about this most of the time. I'm thinking about, do you meet some random plant out there in the world and fuse gametes, or do you might find to mate with a close relative? All right, so that's a rough sense about what we mean when we look at the extent to when we think about how plants mate. And so that family reunion is more like selfing, where the pollen from an individual might land on itself and mate with itself, versus other species where that would never happen. So in many species, you find plants look pretty, flowers are pretty, they are open to the world to receive pollen from not them, and they often have numerous mechanisms to promote outcrossing, that's to say to prevent selfing, and that can be a chemical form of self-incompatibility. It can be the temporal or spatial separation of male and female function, and other sorts of tricks, separation of the sexes on different plants, other sorts of tricks to say selfing's a bad idea. Yet other species, sometimes very close relatives, go out of their way to almost ensure that they're pollinated by themselves. And so sometimes the plants will lose their self-incompatibility mechanisms. They'll sometimes not even open or hardly open to ensure that they get their own pollen. The distribution of this is somewhat contentious. This is probably the best guess. So you can see here, this is the outcrossing rate. Here's our fake histogram. And you can see that there's a lot of plants that are largely outcrossing, but that you see a non-trivial tick up there with these plants that are predominantly self-fertilizing. And then other ways, as you can imagine, not only will you mate with not yourself or somebody in your own species, but you can even mate with other species or subspecies. So I'm going to talk a little bit about the fact that we see introgressive hybridization in plants quite often, and I'm going to make a little bit of use of hybrids between maize and their wild progenitors. Okay, so as I said, there's all sorts of variation that we care about in the world, and that how plants mate allow us to separate out these patterns. So I'm going to talk about these first three in the next few slides. So I'm just going to say for the macroevolutionary folks here, the evolution of plant selfing has been a model system to understand long-term patterns of diversification rates. Okay, so this is an idea I'm starting with just because I think it's so cool. It was the idea that got me really excited about this problem. It was a problem I did as an undergrad, where you can think about conflict between mom and dad over resource allocation. Okay, so if you think about the yummy part of corn, the endosperm, right, that comes from mom. And the more nutritive tissue you get to begin with, presumably the better off you are. So dad and kids would prefer a lot of that, but mom has many mouths to feed. And so you could imagine that there's this conflict, this tension over how many resources offspring wants, right? And under this mode of gene expression called genomic imprinting, you have the differential of expression of a gene based on if it's inherited maternally or paternally. What we often see is that paternally inherited genes act to acquire lots of resources from mom when expressed, while maternally expressed genes, or genes expressed from maternally inherited, tend to make you somewhat smaller. And so an early prediction we made is that if that's the case, then when you're selfing, the mom is the dad, there should be less conflict between the parents over resource allocation. And when you're outcrossing, there should be more such conflict. And so to get a sense about, okay, do we think that this conflict is driving variation in, is driving variation in endosperm development, we can just do a cross between selfing and outcrossing plants, right? And we'd predict that when the selfing plant was the dad, those seeds should be smaller because they're less used to fighting. And in the reciprocal direction, those seeds should be bigger because now the paternally inherited genome from the outcrosser is ready to suck up all those resources. And so we did a literature review at the time, and it seemed like this idea was broadly supported. And since then, it's been really nice to see, as transcriptome studies have come on, you generally see the upregulation of imprinted genes when you're in outcrossing species relative to selfing species. So this has been a fun prediction that seems to have worked out. Another bit of my early work was talking about the response to selection. As I said, many of us are interested in epistasis or dominance or all these components of genetic variation that aren't traditionally heritable. We think they're really cool, but we kind of don't know what to do with them. But to the extent that they might matter, again, you can inherit them when you're selfing. And the reason why, right, is that when you're a selfing plant, you inherit a bunch of the genome together, right? So you can, all these non-additive effects can sometimes be heritable. And so we thought about this in the context of this broad pattern that we see in nature, which is we see mitochondrial genes move to the nucleus in plants. And so this seems like a complicated scenario and plausibly epistatic in that the gene has to be expressed somewhere. It has to be expressed in the nucleus or somewhere in that, and if you have it in either the mitochondria nor the nucleus, you're done. So we did some theory to get a sense about how, under different scenarios, we would, with this epistatic selection, we'd expect to see the mating system impact how many mitochondrial genes are transferred to the nucleus, and we found, as you might expect from that intuitive description, that more selfing increases the probability that you would see a transfer of these genes, especially when they're beneficial. And then we looked out there in the world, we did a survey of how plants reproduce, and we found indeed that plants that are reproducing predominantly asexually or by selfing transmit more mitochondrial, or move more mitochondrial genes to their nucleus than predominantly outcrossing genes. So this suggests that this is an epistatic problem, and that by selfing, you're kind of coordinating that challenging issue. And so the final idea many of us are interested in, right, is about inbreeding depression and how it may or may not be beneficial to mate with close relatives. George was showing me a cool paper he did on this about how the optimal mating distance might depend on genetic variation within a species. And so we think there's two conflicting pressures, right? One thing is you think when you're mating with a close relative, you're more likely to expose deleterious mutations. And indeed, what we find here in 2 years of a field study led by my postdoc is we find that selfed seed has lower survival, consistent with the idea of inbreeding depression. But on the other hand, self-seed keeps combinations together and exposes less, or keeps good things together, and so we think that might coordinate resistance to herbivory, and so here we see less herbivory with more selfing, and that might have to do with the profile of defensive compounds in these plants. Okay. So that's the broad overview about why we care about mating system, how we can expose some of these bigger questions by leveraging the extent to which plants self or they outcross. And so now I'm going to talk about work that was largely led by my first postdoc in my lab, Dina Grossenbacher, about how the patterns of mating can impact where we find plants. And so the first thing is just revisiting this classic idea about where we should expect to see plants, and should we expect to see more selfing plants on islands. The second thing is to say, from looking at islands, how do we expect this to impact the continental range of plants? And finally, thinking about, does selfing allow species to coexist that couldn't otherwise? And so, yeah, Dina's the true hero of this story. So we're going to start with this idea of Baker's law. It's pretty straightforward. You don't need to be a particularly strong evolutionary theorists to grab this concept. The basic idea is, you can imagine you have this plant grows up on this island and it can self-fertilize. This plant grows up on this island and it can't. Obviously, this plant will set seed and be happy. This one won't. So the obvious prediction is that you'd see islands enriched for plants that are capable of self-fertilization. And so this was an idea that seemed intuitive, and there were lots of anecdata supporting that this might happen. And Baker, who's, a famous botanist, said this is totally a thing. But there's very little evidence for this. What you find is somebody looking at their favorite taxon and showing a small number of species that seem to address this. And in fact, this was somewhat contentious. Carlquist here, who's another, big botanist dude, said, I don't think the data stand out very much. He found all sorts of counterexamples, and so this was a fight between old dudes over biology things. Okay, so why do we care? Is it just setting straight this old fight between people? No, right? I think the reason why we care about this is we want to know the extent to which, allele effects matter, the extent to which propagule pressure impacts where we find organisms, et cetera. And so we want to try to do a better job of answering this question. And so this was, the results are simple, but it was a lot of work. So I want to show that there was lots of QC. Dina and Emma and Jeremiah and Boris and other people worked really hard, is all I want to say. So that was a lot. So, thanks them. And the data we ended up collecting looked like this. After a bunch of QC, we would have, plants for a family and look at their occurrences in islands and mainlands. And you can see from this, I forget what they call these sort of plots, but you can see that there's more, that there's an enrichment for self-compatible plants on islands. right? And you can see this in the Asteraceae, you can see this in the Brassicaceae, and you can see this in the Solanaceae, and you can probably see this everywhere. These are the ones we looked at. Interesting tidbit is that the mechanisms of self-incompatibility in these groups are somewhat different, and so it seems to not be an issue that's specific to the mechanism of self-incompatibility. So we find the strong statistical support for Baker's law. We find that islands are indeed enriched for plants capable of self-fertilization, but this doesn't seem to be, a hard and fast rule. There's a reason that Carl Quist might have been skeptical, right? In each of these cases, you find a non-trivial portion here, and so I think that while that says that in that top right, so while this says that propagular pressure and allele effects might have something to do with establishing on islands, there's other features that we might want to look into. Okay. So the intuitive idea that plants can do that selfing plants can do can colonize one alone seems to be supported. It seems to do something. If we're not like super into island, biota, what does this tell us about the rest of the world? So what we did next is we looked at the range size of a bunch of different selfing outcrossing species pairs and tried to get a sense, okay, so now that we're on land, could the way that you reproduce impact your range? And this is an idea that really could have gone either way, in my view, like before we started this thing, because, okay, there's this intuitive idea that by selfing you can make it, you might have a bigger range, but there's all these other ideas that selfers aren't particularly good at being plants. They have higher inbreeding depression. They have a higher genetic load. They can't escape parasites or environmental challenges, so they might have contracted ranges. So we didn't quite know before we started. And the data, at least to my mind, seemed mixed. So this was a question that I thought was a lot more in the air before we answered it. And so what we did is we looked at selfing outcrossing species pairs, and in nearly every genus, the selfing species had larger ranges than their outcrossing relatives. So that's to say, it seems like if you're selfing, you can have a larger range than your outcrossing relative. Now, I'm not certain how to interpret this. I'll tell you this next bit of result here is that we find that the distance, the range size difference increases with divergence time. So the size of those points are about, the posterior probability that these are sisters. But we find that the difference in the range size increases as selfers as the split is older. And so the next thing we find too is that the selfers occur at higher latitudes. And so I can't tell if we wait long enough if it turns out the selfers will have shorter ranges and what's happening, smaller ranges. And what we see right now is that we're seeing that we're in a non-equilibrial scenario since the last, glacial maxima, and now selfers have been able to colonize and spread out, and go to further latitudes, and that's transient, or if we waited till a longer time, if we continue to see this pattern. But it seems like how you mate has a lot to do with where you live, or how broadly you live, or where we find you, etc. Okay, so Dina had these two great results. And the final idea she had was looking at, or that we developed, was looking at when you'd find two species in the same place. And there was a relatively simple motivation here, right? If you have a selfing species at the same place as an outcrossing species, they won't necessarily be competing for pollinators. They won't be, displacing one another with each other's pollen, etc. So maybe they could coexist more. And this has been an idea that Levin suggested a while ago, and that was floating around the literature, and it was supported by a dissertation chapter Dina had looking specifically at a mimulus species pair. This wasn't with me, but this was some of the motivation, where she found that in sympatry they had more divergence in floral traits, but not in ecological traits, suggesting that maybe how you mate might have to do with if you can coexist. So we found 98 sister pairs from 20 genera and 15 families, and we tried to evaluate this hypothesis. And it turns out that this was there was no support. So the extent of range overlap had nothing to, between outcrossing pairs, selfing outcrossing pairs, selfing pairs. It seemed to have no impact. So I'm trying to resolve in my head what's going on here, right? Dina found a cool story here. I'm actually working on another case in Clark here where we find something quite similar, but yet when you do a larger scale meta-analysis, this doesn't pan out. And so it's not, maybe, I mean, my working idea, right, is that species figure out a way to coexist in sympatry, and it can be by this process of mating system, but that might not be a predominant mode. Okay. Are we okay? Yes? 1, 2, yay? Okay. Excellent. So that last bit hopefully got us thinking about what happens when species come together and how the mating system might have something to do with that. And so now we're going to look at patterns of gene flow across the genome and patterns of hybridization when we get two species close together, working particularly on selfing out crossing species pairs. And there's been a burst of sequence data and non-model systems in the past few years, and so we can make some generalizations about what we think is happening when species come together, right? And I think this is your 2-minute summary of what we found here. Gene flow between close relatives is common. For maybe the graduate students in the room, this isn't surprising. You've seen this in every paper, but to me, as growing up, and maybe, I don't know, be interesting to see a distribution by age about if this is a surprising result, but I find this surprising. Nearly, whenever you do some ABBA test or something, we just see so much gene flow. It's wild. But species, despite this gene flow, species often maintain some semblance of identity. Seems to be a second result. And I think we get two different patterns, and I don't know how common they are. It seems like we either see genome-wide selection against introgression, so any road in the genome can't introgress, it's always bad, or you see a few regions often tied up in inversions that have trouble going through species boundaries, and those regions are selected against. And then we even find a few cases where adaptive introgression occurs. So that's my rough sense about what I think this field has shown us. And so the question is, why do we get these results, and how do we lead to one versus the other, and what's going on? Okay. So the first thing I'm going to tell you is just that gene flow is also common in self-rearing outcrossing pairs. We'll see that. It seems to be unidirectional. You'll see that in a minute. And then we're trying to see this big question, which I'm not going to answer today, but if my talk does any use for anyone today, I would like somebody to answer this question one day. So I feel like we're seeing that genome-wide we're selecting against introgression, but I think we have no idea why or how or what the basis is. So just think about that as you go. And I'm hoping that what we can do with our selfing out crossing species pairs can kind of get us there. Okay, so I'm going to first show you what we see with hybridization introgression in the mating system with 3 plants and one other fun organism I've looked at. And so the first thing I'll do, just 'cause I think this is really cute, is to show you how we can find introgression from selfers to outcrossers. This is one of my favorite things because you just don't have to be a great population geneticist to get it. And I think lots of the inference programs, lots of the inference methods we use to learn about introgression are a little strange and get weird. I think this is really nice. So what we've done here is this is Mimulus nasutus. This is a selfing species. This is its close relative, Mimulus guttatus. I've colored all pairwise comparisons between each nasutus sample in when they're very closely related to one another. So you can see that this sample seems to be close to this one in this region. And then there's this region where it seems to be different from everybody. Okay, so that's what this means. And so you can see that as we think, because selfers are depauperate for genetic variation, you see that for most of the genome, 2 selfing individuals seem to be somewhat closely related. But when we look at our outcrossing samples, sympatric with these selfers, we find large genomic chunks that you can see by eye, where they seem to be also identical by descent with the selfer. When you look in allopatry, you don't see that. Another really fun way to look at this result is you can look at percent sequence similarity, and you can see, in allopatry, you see this nice, unimodal distribution, while in sympatry, you see this bimodal distribution, where you see some regions of the genome that are really closely related between species, suggesting that they've recently introgressed across species boundaries. So we clearly see evidence for introgression in this species pair. This is another selfing outcross. Oh, I'm sorry. And what we see is that we see that the selfing genome gets into the into the outcrossing background, and we see less evidence for introgression in the other direction. We see a really similar pattern here in Capsella and two Capsella sister species. So we find that the Greek samples that are sympatric with their selfing relative are more closely related to that selfing relative than are the outer Greek samples. And we can add up the bits of the genome that seem to be recently introgressed, and we can add those all up, and it seems like about 4-point-something% of the genome is recently IBD between an outcrosser and a random selfer. The cute thing is that if you do some algebra about genome-wide divergence from these sorts of box plots, you get almost the exact same estimate, again, saying that we think we're estimating this pretty well. So we think that looking at these chunks is a really nice way to identify recent introgression. So those are two stories where we've looked closely across the genome and found introgression from the selfing species into the outcrossing species. We've recently done a broad meta-analysis where the data aren't as clean and more anecdotal, experts in the field, suggesting what they find or, using, smaller number of markers. But we see that we find gene flow we find lots of gene flow between Si and Sc species pairs. And I think, maybe it's the next slide. But we see that this is largely asymmetric. These are not plants. These are cave fish. But in my look into the non-plant world, we found also plenty of gene flow in this system. I think it's somewhat like a selfing outcrossing species in that these blind cave fish have many characteristics that are comparable to selfing species. They have the proper genetic variation. They seem to have an accumulation of putatively deleterious mutation, more inbreeding, et cetera, and we see very similar patterns. Okay. So what are we selecting when we select against introgression? Does anyone have any ideas? Okay, that's a good one. Incompatibility. Does anyone have better ideas? There must be another option. Local adaptation. Sweet. Anyone have another idea? Or does just George want to do them all? Okay, let's go with those two. I've got others, but we'll start there. So this is George's. This is the idea that we might have. I'm actually quite surprised that we select against incompatibilities. I thought that they would be in sympatry. You start exchanging genes, you would rapidly lose incompatibilities because they're removed by natural selection. Yet in our Capsella example, we see that the chromosome arm where there's a derived incompatibility in grandiflora, we see more introgression of rubella in there. While on the chromosome arm where rubella has the derived partner for that incompatibility, we see less introgression. So it might be that incompatibilities do drive to some extent the extent of introgression genome-wide. So you might find something special at a given locus. The other option is you might find genome-wide for a reason either because there's a million incompatibilities or because there's polygenic local adaptation or because maybe the effective population size of the two species are different such that there's genes that are mildly deleterious and can make it to moderate frequency in one species but can't, let's call it the selfer, but can't in the outcrosser. You might expect to see genome-wide selection against such alleles. And if that's the case, we wouldn't find any specific region would stand out, but we might expect broad genome-wide correlates, okay? And so to evaluate these ideas, what we did is we said, okay, if everything matters, right, everything has some probability of mattering, then the more likely you are to be close to something that matters, the less likely you are to go between species boundaries. And so one way you can be close to something is by having a lower recombination rate. And so we looked at the genome, we looked across the genome in Windows and looked at the recombination rate and said, how diverged are two samples? Okay? And we found when we did this within the pseudus, there was no effect of the recombination rate on genome-wide differentiation. When we did this within Gutatus, we found no effect. When we did this between allopatric samples, we found no effect. But in sympatry, that's when they can possibly exchange genes, we found that there was more divergence with lower recombination, suggesting that regions where if you're likely to be near something in genetic distance, that is, you're likely to be more rapidly removed by selection. So genome-wide evidence for this. We looked at the similar question in Capsella. Here we looked not just at the recombination rate, where we found a similar result, so more recombination, more admixture, but we also found that the more genes, the higher gene density, the less admixture. Again, genes are likely to matter. So if you're in a region of the genome with greater gene density, you have a less admixture or less local introgression. Okay, so we found both these extremes, both these options about what's doing this. We still don't know what's driving this pattern, and I think this is a big problem in the field that I would like to see addressed. So it could be that we have pervasive incompatibilities, right? Maybe this one I showed you here is one that you can map in the lab when you're doing your QTL mapping, but it could be that there are a large number of such incompatibilities that you just don't get to see. Or it could be because there's pervasive local adaptation driven by a large number of small effect alleles, or it could be this idea of the effective population sizes or the differences in how the recessive load is exposed based on if you're selfing or outcrossing. And so I think this is a big question, and I would like to answer it, or I'd like one of you to answer it. I think this is something we'd like to figure out in the next bit. In trying to do this, I ran into this paper by Molly Schumer, and John Poole had a similar paper where they went through this simple logic where they said, oh, I know one way you can see if there's incompatibilities. Maybe you'd find that ancestry from the same species was more often found together than chance on different chromosomes, right? So if I look on two different chromosomes and I see, oh man, I find LD between alleles from the pseudus, then it's like, oh, maybe it's important that they're together, and if they're not together, something bad happens. And so Molly found a whole bunch of what we'd call LD-LD, long-distance linkage disequilibrium, across the genome in her Xiphophorus fishes. John Poole found similar results in Drosophila, and so they argued that that there might just be a lot of these incompatibilities. And so I thought that was a fun idea, but then I thought for a minute about what might be happening here, right? And if you remember from well, there's two simple things that mean that this result does not say what they think it says. So there's lots of evolutionary processes that generate this long-distance linkage disequilibrium. So the two things that came to my mind are, if you remember from intro evolution class, linkage disequilibrium doesn't go down to 0 every generation, even at unlinked loci. It just decreases by a half. So if you start with two different species and start exchanging genes, you're going to find long-distance linkage disequilibrium, even if nothing interesting is happening, so long as you've started hybridizing in finite time. The second thing that can generate this pattern is, imagine some individuals that have more ancestry from species A, others more from species B. You're going to find on different chromosomes that those alleles are going to come together. And the final bit is epistatic selection should act to remove such incompatibilities from the population. So if they were strong, you shouldn't expect to see them anyway. And so I've tried to address this, right? You can point out problems in somebody's work, but Molly and I tried to figure out how to solve it, and I haven't gotten far. So again, if any of you can solve this problem, I'd be very excited. So here's different ways to deal with this problem under different models of hybridization. You can just take the raw Pearson correlation. And this line is the nominal alpha of 0.05, like our traditional significance bounds, and you can see that you have way more significant results than you should when you just do a Pearson correlation. I shade the colors by if you find like ancestry together or like ancestry apart, and so you can see that these sort of traditional correlations have a huge excess of finding positive linkage disequilibrium by ancestry. So you find alleles from the same species on different chromosomes, together. Second thing we thought about was doing a partial correlation, so regressing out genome-wide admixture proportion, and that overcorrects, doesn't solve the problem. There's another thing that doesn't work. Basically, we don't know how to do this. Good statisticians should figure this out. Okay, so we've learned that the mating system can impact where we find species, and at least we find that there's some gene flow between plants and different mating systems. Oh no, did I really lose that one? Okay, we'll tell you more in a minute about this thing. But I'm going to talk a little bit about now what happens when you start exchanging genes between species and some consequences for the genome of our species. Okay. And particularly how we can see tensions over mating resolve in this process of gene flow. So I'm going to focus on corn, which is something I learned is a beautiful plant that has lots of really cool biology. It's not just, fuel and plastic and things that we eat. They're really pretty, and they have really cool biology and really nice natural history. And one of the cool things, and I don't know if you see this for too many other species that we eat, but that you find them sympatric with their wild progenitors. And so Zea mays, you might know as corn, but there's, a few subspecies of Zea mays. And in the highlands of Mexico, you see Zea mays mexicana, sympatric with what they call maize landraces that are farmed traditionally. And what you'll see is that you find, for example here, in our highland maize populations, you find a non-trivial portion of their genome recently derived from Mexicana. Okay? So often, right, we see species. Species seem to be things that are good, like species don't fuse often. So why don't they, right? Presumably their hybrids aren't particularly good. And so there's usually thought to be some evolutionary pressure to prevent mating with the wrong species, right? In a process known as reinforcement. We were curious about how this might work in maize and teosinte, and we were motivated by these gametophytic factors. So these gametophytic factors act as follows. So if you think about a plant stigma, right? You have these pollen that land on it. If your pistil doesn't have a functional, I don't know what we call it, a rejection allele, let's say if it's smallest genotype, it can be pollinated by any pollen that lands on it. By contrast, if you have let's call it a choosy pistil, it will reject pollen that can't overcome this barrier. These gametophytic factors, TCB1 is one example. There's also two others. Let's call them TCB2 and TCB3. It's not right, but there's three such loci pairs across the genome, and they act in this somewhat close to this simple cartoon way. And something that's weird that we see when we look at the natural frequency of these things, and something that got us a little confused for a little bit, is that you see that perhaps not surprisingly, when teosinte is found with maize, you find that they have a lot of these, let's call them choosy pistil alleles. That's great, that makes sense. They're trying to reject maize pollen. Fortunately, it seems to not do them much good, because for some reason, while throughout most of maize's range, they have this non-competitive, pollen. When they're sympatric with maize, they have pollen that can overcome teosinte's stylar barrier. So it seems like teosinte is trying to avoid pollination by maize, but all for nothing. And so how and why could that be? We're developing some theory to think about this right now, and then we're doing some sequencing to look into this idea. And so the simple model is that there could be a sexual conflict over reinforcement. That's to say, teosinte styles do best to avoid mating with maize. Conditional on the maize pollen landing on the style, it's better to successfully pollinate that than to do nothing at all. And so we we started with what we could think of as the simplest three-locus model to see if these ideas make sense. We have a locally adaptive allele linked to this, to such a gametophytic factor. And so we start with a non-zero frequency of a very rare rejecting alleles, pollen rejecting allele in teosinte, and a somewhat modest frequency of the pollen allele that can overcome this barrier in teosinte, while in maize we have neither such alleles present, which roughly characterizes what we see in nature. Okay, and then we go from genotype frequencies, we migrate by pollen, we mate according to some rules, we make some genotypes, we do some selection, and we do that for a bunch of generations. And this is one sort of cartoon of what we see happens, is that we see that, we see this reinforcement evolve. So after some number of generations, we see this big F allele, or here it's on this big M F haplotype, spreads through teosinte because they avoid mating with maize. But once that thing gets off the ground, the big M allele adaptively spreads through maize to allow it to overcome this barrier. And so that's our prediction. The theory is pretty fun. We can mess around with, recombination rates, the strength of selection we need, the number of loci in the genetic architecture of these incompatibilities. And we generally see that so long as selection is sufficiently strong, or local adaptation is sufficiently strong, we get this pattern. We can also think about what allows for strong or weak reinforcement to arise, and with high selection you get more reinforcement, and with low selective coefficients, the maximum reinforcement that you hit gets higher with more migration, just because you have more things to select against. Okay, so I've been really excited about this result because I feel like it explains a pattern in nature that got my head, got me a little confused for a little bit. It gives us really nice predictions. So we're currently taking a bunch of maize and teosinte plants, trying to cross them, and getting a sense about can they accept or reject one another's pollen. And then we have the strong prediction that the maize that can pollinate teosinte have recently and adaptively introgressed teosinte pollen alleles at these loci. So we're excited to test this out. Okay. Thanks all for bearing with me. This is like one last something story, and then the last one's like a slide, and then we'll be okay. Okay, but this one's hard, so bear with me. This has been a bit of a confusing problem that I've really enjoyed thinking about. Another somewhat simple evolutionary puzzle. But we're just learning more and more about how plants can reject pollen, and it turns out that in the system that we've known about for the longest time, that we see in most of angiosperms, the solanaceous space, what they call non-self collaborative incompatibility, we found that's under long-term balancing selection, we found this weird mechanism, we being the field of evolution, not me, of collaborative non-self recognition. So here's how it works. As opposed to the simplest models of self-incompatibility where you say, oh, this isn't me, you're cool, where then all you need is one new mutation to allow self-incompatibility. This non-self system is a little bit more confusing. So you can think about it as a lock and key or a poison and an antidote. What happens is that the styler part has, let's say, a lock, and pollen then has kind of a chain of keys. And if any of those keys, those keys are called SLFs, and the lock is called an sRNase. If any of those keys can unlock that styler barrier, that cross is compatible. If they don't have such a key, that cross is incompatible. And so the way you become self-incompatible is by not having the key to your own lock. And so this is kind of a hard problem, right? Because how do you get a new self-incompatibility? We have 40 such alleles in nature, 70. We have a relatively large number of such alleles in nature. They're conserved for a long time, but how can one show up, right? Without a new lock, the key is useless. Without a key, the lock is pollen limited. So the general model is you get a new key, you get a new lock. So now there's something can pollinate that lock. That key invades, and then gene conversion copies that key from one haplotype to another until all of a sudden there's a new locus here. So that was a verbal model. We wanted to turn this into something a little bit more rigorous to see if it worked out. And so we started with lucky haplotypes that have the key to the new lock, unlucky ones that don't. You're fertile if you can accept pollen, and then the only way you can change here is by gene conversion. Okay, so the good news is this basic model starts to work. You can get a new lock, you can get a new key. This is work that Alex led. I found it to be really fun. But the problem is that once you have some haplotypes that can fertilize more styles than others, those are off to the races, and they're ready to replace all the other haplotypes in the population. So while this process of a new key, a new lock, and then gene conversion from one haplotype to another can generate a new S allele, it can also lead to the demise of a bunch of S alleles. And so it's not guaranteed to be a diversifying process. And so we have to think about how this can happen. And so the model we turned to was a lot like that of evolutionary rescue. So if you're familiar with the evolutionary rescue evolutionary rescue model, you have a population that's not well suited to its current environment, and so it has it shrinks more than it grows, and you have to rescue that population by a new mutation that has to show up and allow it to have a positive growth rate before it goes extinct. And here the analogy is really similar, right? Except it has to happen numerous times, numerous haplotypes, and then it has to move from one to another by gene conversion. So with a little bit of a few math tricks, Alex worked through these rescue probabilities, and as you have higher gene conversion rates, you're more likely to rescue all haplotypes. As there's fewer haplotypes to begin, you're more likely to rescue more haplotypes. And as the key initially exists on fewer haplotypes, you're more likely to rescue all of them. So we can find some broad parameters here, and we can take these results that I've shown you here, and we can do a simple Markov chain to say what's the equilibrium number of S haplotypes. It turns out that a key component is the population rate of gene conversion, but that we can get, with relatively high rates of gene conversion, we can get a number of S haplotypes comparable to those that we see in the world. And so if this model is to be proved true, we have to argue that there's a relatively high input of gene conversion between S haplotypes. If not, we're still kind of screwed, and we still don't know, where and how we get the large number of S haplotypes that we see in nature in these collaborative collaborative non-self incompatibility. Okay. So what we learn from this is that learning about biology matters. How, in a case where we have the simple, model of how many S alleles we'll see, Wright did some simple math to show that you should have a lot, but that thinking about the process makes it harder, and it leads to something that's quite fun. And so this final thing that I think is really cute is an easier way to get new S alleles is just to borrow one from another population. And so we showed that we did a little bit of theory to show that that even with strong selection against hybrids, you can have the adaptive introgression of S haplotypes across species boundaries. And it seems like self-incompatible taxa are enriched for high amounts of gene flow, plausibly because we favor moving S alleles across backgrounds. We should look at this more carefully by perhaps asking if these S alleles are moving between these species. In summary, I hope that I've somewhat convinced you that even if we don't care about how plants mate, it can really get to some of these cool questions, and that allows us to dig through our understanding of evolution and all these higher level questions. And then I'd like to thank everyone here for the invitation and attention. It was really nice to chat with you all today. And I'd like to thank my mentors, collaborators, postdocs, students, family, and money. All right, thanks a lot, and I'll take any questions. Anyone, anything? Yeah, Tim.
In your study of selfing, in your study of selfing, you kind of have this huge group called selfing, and I'm wondering if you sort of compared aplomix to selfers in terms of their distributions, Baker's law.
Yeah.
Do they behave different?
We did, and I think we decided we lacked any power to do anything serious. So it would be, so the question, right, is there's plants that can self versus those that might that are asexual, is there a difference there? And I think we didn't have any power, but I forget. Why do you ask? What's that? Was there another one, or should we?
If I could just follow
Yeah.
up then, but some of them have mixed mating systems too. But did you parse that out?
Yes, that was also. So this is one of these working groups, and we kind of pulled our hair for a bit and thought about a million ways to cut this up, and we thought about, should we look if we're closer, if the island is closer or further from from the mainland, and should we look about whatever. We had trouble coming to agreements about what to do, and this seemed to be the thing in the end. I forget if we didn't analyze the self. Oh, yes, I know the answer now. Most people don't know, so there's lots of data about if you can self or not. Finding the rate of selfing is a much harder problem where there wasn't enough data to do much. Yeah, sir?
I think I have the same question. There's a lot of different activities for tropical trees which have varied mating systems. Sometimes you could say that a tree or a species is an obligate outcrosser because they're separated. You can rarely, I've never seen a case where you could say it's obligate for a self. It's always, people will say it's always a mixed mating system. So there's never just a selfing system. So I was wondering, it's a similar question. If you take that into account, there's not a dichotomy between selfing versus outcrossing. What do you get there?
Yeah, so again, we just had these sort of probabilities so basically to say, we said, are you self-compatible or not? And that was if you go to the greenhouse and you do a bunch of crosses, you do a bunch of self, is roughly, is that rate, better or worse than 80% of the outcrossing rate or whatever in terms of seed set. So for that doesn't matter. And also in terms of Baker's law, we hemmed and hawed about maybe all you need is the ability to set seed, or maybe you need the ability to set seed, in the absence of pollinators, or maybe you need the ability to set seed if there's no other plant there, So we're thinking a little bit about how to think through those ideas, and it's not clear. Well, I think that there's probably no obligately selfing plants except for clystogamous ones that never open. It does feel like there's if you just go to that early histogram, right? It seems like there is a middle where there are some plants that have a mixed mating system, but it seems like it's not it's not unimodal. It's not even uniform. It seems like those edges are enriched for things, and there's obviously the Landy and Shemsky paper made that strong prediction. So maybe we're all too happy to see that in the data, and maybe it's not there as much as we think. But how mixed maters can even persist is a complicated question in evolution. Yeah, Gina?
So, on your recombination rate work, can you sort of comment, do you think that recombination rate is a phenotype that can evolve given the presence of closely related species? So is it the community context can influence evolution recombination rate itself?
Yeah. That's a great question. I would say it could, but probably a bigger concern or a bigger issue. Well, so there's a few ways you could Okay, sorry, there's a lot there. That's a great question. Let's see if I can answer all of it. One thing you can have happen is you can imagine local changes in the recombination rate. So there's some theory to say in the space of gene flow you might evolve inversions or a locally lowered recombination rate to decrease that migration load. So that's one thing that could happen. The other thing that I think needs to be considered, that I need to think about harder, and maybe the field does, is that in general, recombination works worse when you're less homologous. And so maybe even without any adaptation or any sort of genetic change in the recombination rate, it might be that those maps I tell you there might not be correct because we're usually making a map within species, and it might be that when you're hybridizing with something more distant, that might not be representative of the genetic map within species that we see. And so I think in addition to plausibly having some evolution of the recombination rate, I think just this plastic change in the recombination rate that happens automatically by mating with something different is another thing that we should probably think harder about, and I'm starting to. Anything else? Well, thanks so much, everybody. Appreciate the day.